Hello students, welcome to chapter 12. We're going to be talking about genes and inheritance. Basically the history of how science has figured out inheritance and how they figured out the method of inheritance and everything. So first of all, we have the beginning of genetics and everything, and this kind of began with Gregor Mendel. So in 1860s, Gregor Mendel studied the patterns of inheritance by experimenting with garden peas. So average peas through their flowers, through their fruits, through their the way that they grew, everything like that. But until about the 1900s, most scientists believed in the blending theory of inheritance. So this meant that you had the mother and the father, whatever traits that they had blended together to make the offspring. Gregor Mendel, whenever he published his works, kind of went against that blending theory. So he studies specific characters, specific traits that had alternative forms that went against that blending theory. So his model organism was Pisum sativum, which is the garden pea. So these peas can self-fertilize. They can self-pollinate. Basically, you have the male and the female parts on one plant, and they can pollinate themselves. But Mendel prevented that from happening, basically by cutting off the anthers, cutting off the pollen-producing parts, so that they don't self-pollinate. So that they can cross-pollinate. So one will be pollinating the other one. So to begin, Mendel chose the pea plants that were true breeding. In other words, they had those traits that were non-heterozygote. They had to have both dominant and both recessive. So part of Mendel's conclusions, Mendel established that characters were passed on to the offspring through discrete hereditary factors. And these hereditary factors were through, later on called genes, right? And these genes were later on encoded into DNA whenever they found out what DNA was in molecular form. Adult plants carry a pair of factors, otherwise called an allele. So alleles are different forms of that same gene. So you can have one form of the same gene on one chromosome and one form of the same gene on a different chromosome, right? So if you have a diploid, you can have a yellow or you can have a green of that same form. Let's say in a matter of, hitchhiker's thumb, you can have not hitchhiker's thumb or yes hitchhiker's thumb. That's a binary system. If an individual pair of genes consists of different alleles, you have one dominant and then one recessive, right? So that's your binary, one dominant or one recessive. Pairs of alleles segregate as gametes are formed, right? So through meiosis, you have those alleles that segregate whenever those gametes are formed. So meiosis occurs, gametes are formed through haploid segregation. And this is through what's called Mendel's principle of segregation. So one allele has half the gametes, and the other allele has, or one gamete has half the alleles, and the other gamete has the other half of those alleles. So we have some genetic terms. Homozygote, meaning that you have a true breeding individual, right? This is kind of what I was explaining before. True breeding, both alleles of a gene are the same. Okay? In a diploid organism, you have two sets of chromosomes, right? So if you have two sets of chromosomes, both alleles are going to be the exact same type of allele. So both genes are going to have the exact same type of allele. So it's either going to be dominant-dominant, or if it's going to be recessive-recessive. In this case, it'll only produce one type of gamete in that type of gene, right? It'll only produce one type of gamete with that gene. A heterozygote is an individual with two different types of a gene, right? In the case of the diploid, you'll have a dominant and a recessive. Produces two types of gametes, half having one allele and half having the other allele. So half having a dominant and half having recessive. We have monohybrid. An F1 heterozygote producing a cross that involves a single character. So the F1, meaning that that's the offspring from the parent. Okay, so the parent will have an offspring. The offspring is called F1. That's the first, what's called the first filial. Okay, first filial heterozygote producing a cross that involves a single character. So a single character, meaning that's the single trait. Okay. Monohybrid just means that it's one trait. If you say it's a monohybrid cross, that's two individuals that are crossing, right? So cross between two individuals that are each heterozygous for the same trait. Okay? The reason that they're heterozygous, all right, is because you need to find out how each of these are going to be in the second filial generation. All right, and I kind of will show you further on in this PowerPoint how that's going to go whenever we talk about Mendel's crosses. Okay, so heterozygote produced from a cross, that's a monohybrid. And if you cross those two monohybrids, right, that's just a monohybrid cross. And that's an important concept when it comes to one of Mendel's studies. Okay. A dihybrid means that this involves two characters, two traits. Okay. Heterozygous for each of those traits. Each of those pairs of alleles, two genes involved. Dihybrid, that's a cross between, so if you have a dihybrid cross, that's two individuals that are heterozygous for two genes. Genotype and phenotype. Genotype refers to the genetic constitution of an organism. Basically, it's genes and alleles. All right? So, if I told you the genotype of something, that would basically be the illustration of its alleles. So, basically, it would be homozygous dominant, heterozygous, or homozygous recessive. Right? Its genotype for homozygous dominant would be large A, large A. Right? Large A, large A. That's its genotype. Phenotype refers to the organism's physical appearance. Okay? So, phenotype of a homozygous dominant, let's say that controls this mouse's fur color. And if this mouse's fur color is controlled by that, it's brown. Right? Kind of a darkish brown. That's its phenotype. Okay. So, two alleles for flower color. P, it's the genotype, and then purple is the phenotype. And then small p is the genotype, and white, small p is the genotype, and white is the phenotype. When we get into the whole idea of alleles, the idea of genetics, we have to talk about probability. So that sperm has a random set of genes, that egg has a random set of genes. Okay, and if you talk about one gene, it's a monohybrid. If you talk about two genes, it's dihybrid. Right? Talk about more than that, you get a lot more probability. So you have the random fertilization of an egg by a sperm. Is the mathematical possibility of an outcome a matter of chance? Certain outcome has a probability of 1. An impossible outcome has a probability of 0. Right? When two or more events are independent, we can calculate the probability that they will occur in succession using what's called a product rule. Basically, we have to multiply the probabilities by each other. You have to multiply their individual probabilities. So, if we have one head and one tail, if you flip each coin individually, let's say that you have the first toss, that's half and half. You get the first toss, that's you get head, half you get tail. Let's say the second toss. What's the probability of you getting a head on the first toss and the second toss? You have to multiply 1/2 by 1/2. What's the probability you get a head again on the third toss? So if you get a head, and a head, that's 1/2 times 1/2 times 1/2. Right? So you have to do that in succession. So if you multiply that over and over and over again, the number keeps going down and down and down. So that's the product rule. You multiply the individual probabilities over and over and over again. So how does this pertain to our probabilities of the Mendel crosses? So the probability of obtaining purple flowers. Let's say that we have 2 ways to get purple flowers. Over here we have our Punnett square. And a Punnett square is basically you have one genotype. You have one genotype for the male and one genotype for the female. So I'm going to write this down. In case you don't know how to do a Punnett square. So we have the male that is heterozygous and the female that is heterozygous. And we're going to cross them. First, what you're going to do is separate the alleles, like so. And then follow each individual gamete across the table. Okay, so big P comes down, and big P comes down. Little p comes down, little p comes down. Big P comes across there, across, and across there. So each of these represents a gamete, okay, that each of these separated into. And this is how you do a Punnett square. So each gamete has a half of a probability to give to each offspring. So if each gamete gives half probability to give to each offspring, that means that 1/2 times 1/2 will give you 1/4 a chance to give you one purple dominant, otherwise 1/4 ability to give you homozygous dominant purple flower. 1/2 times 1/2, 1/4 ability to give you a homozygous dominant flower. How much would it be to give you a heterozygous flower? Well, if it's 1/4, it'd have to be 1/4 plus 1/4, right? Because you have 2 1/4s here. 1/4 plus 1/4, that's 2/4, that's 1/2. So that's 1/2 heterozygous probability. And then homozygous recessive, that's 1/4, right? Because 1/2 times 1/2 is 1/4. So that's your probability. And I know this kind of seems easy right now because you can just kind of see it. You can just see, yeah, that's 1/4. Yeah, that's 1/2, that's 1/4. But when you get into dihybrid crosses, this is going to be a lot harder to see. It's going to be a lot harder to try and understand. And if you can mathematically think about this, if you can mathematically work this out, it's going to be a lot easier for you to understand how this works. So if this is 1/4, you can calculate this into percentages. So what is the percentage likely that you're going to get homozygous dominant? It's going to be 25%, right? Because 1/4 is 0.25 times 100, that's 25. 25%. And then heterozygous, that's 50%. And homozygous recessive, that's 25%. That way, whenever you're calculating what's called genotype frequencies and phenotype frequencies, you can say, what is the genotype frequency of homozygous dominant? The genotype frequency of homozygous dominant is 25%. And that's how you say that. So the genotype frequency of heterozygous is 50%. And you can guess the other one. So for your attendance here, what I want you to do is tell me what the phenotype frequency is for this box. For this box on slide number 9. Tell me what the phenotype frequency is for this specific one. So we have probability in Mendel's crosses. What is the phenotype frequency? So we talk about test crosses. So geneticists can use test crosses to determine whether an individual with a dominant trait is heterozygous or homozygous. So whenever you look at an individual, whenever you look at a specific organism, sometimes you're not going to be able to tell if it's heterozygous or homozygous, right? Because the heterozygous is going to have a dominant trait that's going to overlook that recessive trait, right? It's going to cover up that recessive trait. So sometimes what you need is called a test cross. And that test cross is a cross between an individual with a dominant phenotype and a homozygous recessive phenotype. All right? So, homozygous recessive individual, why would you need a homozygous recessive individual there? Why wouldn't you use any other type? Well, it's exactly what it says right here. So, if you used a homozygous recessive individual, what would happen if you had a dominant trait, a homozygous dominant trait? You would have all of the predictions, all of the offspring come out to be only dominants. There would be no recessives. But if you had a heterozygote individual, if you test crossed and you found out that you crossed your dominant phenotype with a recessive, and then your offspring came out, and it's half of your offspring came out with recessive traits, then you would find out that your dominant phenotype is actually heterozygous. Your dominant phenotype parent is heterozygous. Do you see how that happens? Because if it's heterozygous, it's carrying a recessive trait, right? And the recessive phenotype is pulling out that recessive trait and giving it to the offspring. However, if it was a dominant phenotype, it wouldn't be able to pull anything out because there would be no recessive phenotype there in order to make any more recessive offspring. Other crosses that Mendel led. So this is called a dihybrid cross. Okay. Mendel crossed a true breeding plant with round yellow seeds. So one of the traits was round and one of the traits was yellow. So one of the traits was its shape, one of the traits was the color of the seed. With a true breeding wrinkled green seed. Okay, so the wrinkled green seed was recessive. And he did this through to the F2 generation. Okay, so he did this the first generation, and he did this to the second generation. Okay. The first generation held the heterozygous filial generation. Okay, the first filial generation held the heterozygous generation. The second generation held a generation with the phenotype ratio of 9 to 3 to 3 to 1. Okay, and I'm going to show you how that goes. Okay, so we have all the gametes on the top, all the gametes on the side here. And then all in the center here are the potential offspring that we can get with these gametes. And I realize that this might be a little bit more difficult for you to see than what's on the PowerPoint. Okay? But we have yellow, unwrinkled, all the way across on the top and all the way down. Okay, that makes 7. Then we have yellow unwrinkled here and yellow unwrinkled here. And that makes 9. And then we have green unwrinkled, and then we have green unwrinkled, and then we have green unwrinkled here, and that makes 3. Then we have yellow and wrinkled, yellow and wrinkled, and yellow and wrinkled, yellow, wrinkled, yellow, wrinkled, yellow, wrinkled. That makes 3, and then we have green, wrinkled, and that makes 1. Okay, that's our phenotypic ratio. Now, this is all genotypic ratio right here. This is all our genotypes. All right. If I were to say the genotype ratio, what would I say? This is 1. This is actually 2 right here. That's 1. That one's, and that one's the same. That one, that one, that one, and that one is the same. That's 4. So that's 2, that's 4. That one's 1. These 2 are the same. These 2 are the same. And that one's the same. So it actually goes 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. The genotypic ratio of this is a lot more difficult than the phenotypic ratio, just because there's a lot more genotypes on here than there are phenotypes. But I want you to keep something in mind. If we were to separate each one of these gametes, or each one of these genes, how do you think it would affect the probability of them being separated versus them being together. It wouldn't affect it at all. And let me tell you something why. 1 to 2, the 1 to 2, the 4 to 2, the 1 to 2 to 1. This probability, 1/16th, 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 has the same exact probability as a monohybrid cross. And this is actually part of the same solution that Mendel had whenever he was doing a dihybrid cross. Okay? Part of the same solution, because what he thought up is called the principle of independent assortment. So, Mendel showed that traits of different characters were distributed to offspring independently. They weren't inherited together. Okay, this is called independent assortment. So if I were to take this entire dihybrid cross and put it in the same probability independently to say that R or Y were separated from each other, and say that R has the same probability as Y, it would have no difference that if I did a dihybrid cross. Now that is to say, I would still have to do what's called a chi-squared test to see if there was a difference between the R gene and the Y gene. If there is a difference between R gene and Y gene, or if they do interact with each other, then I would have to do a dihybrid cross to say, yes, there is an interaction with each other, and yes, they do occur on the same part of the gene, and yes, they do have a linkage point. However, that's for a later time, and we'll be talking about that during another lecture. However, at this time, a dihybrid cross is still important, but know that because of the principle of independent assortment, dihybrid crosses still have the same probability as monohybrid crosses. So the alleles of genes that govern two characteristics or two traits assort independently during formation of gametes. If I took two chromosomes, and they have two alleles on those chromosomes, and they separated during meiosis, right? They would have independent assortment on the gametes depending on where they go. If they have two genes, they would assort independently from each other depending on where they go. Meiosis, which related to Mendel's factors, wasn't discovered until 1890. And then Walter Sutton recognized the similarities between Mendel's genes and the behavior of those chromosomes. Okay, so Sutton discovered those particular sites on the chromosome, which he called the locus. Okay, so a specific part on the chromosome is called a locus. Chromosomes of each pair are separated and delivered singly to the gametes, right? Because they're haploids, as alleles are of a gene. So each gene has an allele. Chromosomes carry one allele of one gene. Separation of a chromosome pair in meiosis is independent of the separation of other pairs. As an independent assortment, of the Mendel's dihybrid crosses. Right? So if each chromosome carries one allele per gene, that means each gamete only carries one allele for one gene. So each gamete carries one allele for one gene. So if you have one sperm carrying one allele and one egg carrying one allele, they'll come together, and then you'll have two alleles when you have the zygote. In fertilization, one member of each chromosome pair is derived from the male parent and one from the female parent, an exact parallel of the two alleles of gene. Sutton correctly concluded that the genes are carried on the chromosomes, known as the chromosome theory of inheritance. Okay, so this figure kind of shows you the similarities between what we know is the behavior in chromosomes in meiosis and the corresponding Mendel's principles of independent assortment. Right? So chromosomes occur in pairs in diploid individuals, right? So we know that meiosis occurs when chromosomes replicate right before meiosis. And then they go through metaphase, they go through the first meiotic division, second meiotic division, and then gametes appear down at the bottom, right? And then at the end we have 4 haploid individuals, each with one set of chromosomes. With Mendel's principles of independent assortment, along with Sutton's chromosome theories of inheritance, we have the same kind of thing happening, where we have pairs of diploid individuals. We have the allele pairs. 2 alleles of a gene segregate from one another during gamete formation. During the segregation of those alleles will pair independently from each other whenever they form the gametes, right? So they'll deliver them singly, deliver whatever they thought was that genetic material singly into those assorted pairs. So it's basically what they thought was the exact same thing that is happening. There are a few things that can modify this principle of inheritance. So further research revealed many variations to this basic principle of dominance and recessive inheritance. There's incomplete dominance, where you have one individual that has a dominance trait and then another individual that has a recessive trait. But that dominant trait doesn't completely hide over the recessive trait. Okay? So it makes it almost blending, but it's not exactly blending. And then there's codominance, where you have 2 dominant traits that kind of force themselves together. Okay, so codominance, one dominant trait and another dominant trait will show together at the same time. There's multiple alleles. So sometimes you'll have multiple alleles on the same trait that will, or the same gene that will show at the same time. There's epistasis that we'll talk about, polygenic inheritance that we can talk about, and then pleiotropy. This one's a little bit more in depth. All right, incomplete dominance. When one allele of a gene is not completely dominant over the other allele. So the phenotype of the heterozygote is intermediate between the phenotype of the dominant and the recessive heterozygotes. So you have a monohybrid cross between the F2 individuals. So you have a 1 to 2 to 1 ratio. You have the homozygous dominant, heterozygous, and the homozygous recessive. So the homozygous dominant in this case is red. The heterozygous is a pink color, and the homozygous recessive is a white color. So the heterozygous is kind of a blend between red and white, where the red doesn't exactly show all the way between, or the red doesn't exactly show all the way red. Codominance occurs when you have an effect of two alleles of a gene that are equally detectable in heterozygous. So sometimes you have what's called two dominant alleles that are just showing out at the same time. Example, what's called human MN blood. This is a glycoprotein in blood types. So sometimes you'll have a genotype LM or LN, and if you mix those two, you have LMN, and it's just two types of glycoprotein found in your blood. Another type of codominance that's a little bit more prevalent because you can see it is these chickens. So chicken with all black feathers and chicken with all white feathers. If you can mix them, you can have a checkered chicken feather. Checkered chicken. All right, then we have multiple alleles. Although an individual that has only two alleles for a gene, you can have one with multiple alleles. This has two different types, more than two different types of alleles in a gene. And this can be representative of a whole population. So this is something that is ABO in our blood, right? So gene B or gene A or gene O in our blood, especially sometimes antibodies, may change in the way that our we may receive blood, we may give blood, or whatever. And this is all inherited by our parents. So multiple alleles of a gene may contain differences at one or more points in our DNA sequences. And this is all in a gene. And if you're curious about that, here's a Punnett square of that situation. So if you have what's called IAA, that's all A type. Or if you have IAIB, that's AB. And if IAI, lowercase I, that's A type. I'm not sure how the antibodies work. I believe that might be something that's a little bit larger than this. But if you have type O, that's recessive. That's all recessive traits. Okay, so you have to actually have it from 2 type O parents. And or you can have it from either a B or an A parent that is a carrier. Okay, another one is epistasis. Epistasis is 2 genes that interact. Basically, you have 2 different genes on the same DNA, and those can interact with each other depending on where they're located. So alleles of a gene at one locus inhibit or mask the effects of alleles of a different gene at a different locus. The result of epistasis is that some expected phenotypes don't appear among the offspring. So sometimes you'll have phenotypes that just don't really show up, or you'll have some phenotypes that show up a little bit different. Okay, so in this case, you'll have these mice over here. So instead of just black and white, you have a brown coat that shows up. Epistasis is an important factor in determining an individual's susceptibility to common diseases, such as insulin resistance. So this is something that can pass on through your offspring. And this is just an interaction of genes affecting another gene. And then there's polygenic inheritance. This is a continuous distribution of phenotypes, typically resulting from polygenic inheritance, which several to many different genes contribute to the same character. This can be height, this can be skin color, this can be hair color. These are quantitative traits, right? So anything that has a very large variability. So when numbers of individuals in a series are defined classes, you can plot it on a graph, and then you can have what's called a bell-shaped curve. And that's kind of what's here on this side. It's a kind of a bell shape. So most of the people in that population are going to be in the middle of that bell shape. Okay? The least amount of people are going to be on the sides. Okay? So in this case, if you have skin pigmentation, most of the people are going to be in the middle area, right? Most of the people are going to be in, or most of that, whatever it's talking about, is going to be in that center area, that center tannish area. The least amount of people are going to be in the whitish area. The other least amount of people are going to be in the darkish area. Or whatever it's talking about. I'm not sure if it's talking about people or if it's talking about fur, or it's not really, that's skin pigmentation. If it's talking about an animal or something. Anyway. When numbers of individual, polygenic inheritance is often modified by the environment. So, in this case, what can happen is if the environment is differentiating, it can be skewed, right? It can be skewed in one direction. So let's say that if you have a quantitative characteristic of skin color and you're more towards the equator, you're going to be more skewed towards a darker color, right? Because it's going to be more towards the sun. But if you are more, or if you're less towards the equator, you're going to be more skewed towards a lighter color, right? And I'm not talking about humans, I'm talking about anything, right? Because the melanin or anything in your skin is going to cause you to want to protect yourself from the sun. All right, the next thing is pleiotropy. Pleiotropy is a single gene that affects more than one character of an organism. So, if you have a single locus on a single gene, it can actually affect more than one trait. It can do more than one characteristic. And one of the things here is sickle cell anemia. One of the traits that it can do is sickle cell, and the other trait it can do is a normal cell. It can switch back and forth, and it's not a very good thing. So, it's caused by a recessive allele that a single gene can alter that hemoglobin. Has a wide range pleiotropic effects that damages many tissues and many organs. So it can switch between sickle cell or normal cell, and it's not a very good thing. This is the end of this. Make sure that you go through and try to find that attendance assignment. All right. If you have any questions, please let me know. Other than that, please go through the lab assignment. Also, next week is the exam number 3. Make sure that you're studying for that. All right. We don't have a day to review for that, so I may put something together, but I may not have time. We will have to see. Anyway, please ask me questions if you need to. All right, through email or through any means necessary, probably through email. All right, please ask me questions. All right, I will see you later.